Good afternoon. Today I would like to talk to you about the effects of blood cells on thrombin generation. So, thrombin generation is most commonly measured in platelet pool plasma. The clinical utility of thrombin generation in PPP has been shown in many clinical studies. Thrombin generation is able to predict both bleeding and thrombosis in certain patient groups. Furthermore, it can be used to monitor replacement therapy in patients with hemophilia or to monitor anticoagulant treatment. Additionally, it is a very useful research tool to explore mechanisms of thrombosis and hemostasis. Measurement of thrombin generation in platelet pool plasma has several advantages compared to the measurements in platelet-rich and whole blood. Samples can be collected and stored for several months or even years, and this allows to batch-wise measure at high throughput. Additionally, several commercial assays are available, and great effort has been made to standardize the assays. However, a major drawback of thrombin generation in platelet pool plasma is the lack of cells. In vivo, most coagulation processes take place on the surface of cells. In platelet pool plasma, artificial phospholipids have to be added to the reaction mixture to mimic the presence of cells. These artificial phospholipids provide the coagulant surface, which in vivo is provided by cells. Therefore, the contribution of an individual's blood cells is not represented in PPP, and therefore it is not possible to study cell-mediated thrombosis or bleeding in platelet pool plasma. First, I will shortly introduce the cell-mediated phases of coagulation. Physiological hemostasis is often represented in three phases in a three-phase cell-mediated model. All phases require the presence of cells or cellular components to establish normal hemostasis. Coagulation is initiated when the endothelium is disrupted, which exposes tissue factor expressing cells in the subendothelium. Circulating factor 7 binds to tissue factor and forms the tissue factor 7A complex, which proteolytically cleaves small amounts of factor 10 and factor 9 into factor 10A and factor 9A. Factor 10A associates with its cofactor factor 5A and forms the prothrombinase complex on tissue factor expressing cells, which converts little amounts of prothrombin into thrombin. During the amplification phase, the sparks of thrombin that were formed during the initiation phase activate platelets that adhere to the site of injury, and this leads to the exposure of phosphatidylserine on the membrane of the platelets. Additionally, factor 11, factor 8, and factor 5 are activated by thrombin, and the tennase and prothrombinase complex are formed on the surface of activated platelets. And finally, during the propagation phase, a burst of thrombin is produced on activated platelets, leading to the formation of sufficient amounts of cross-linked fibrin fibers to form a stable thrombus. All these phases of hemostasis require the presence of cells, and this highlights the importance of adding cells when you measure thrombin generation. Therefore, we think that the presence of cells gives a more insights into hemostasis that is missed when we measure only in platelet pool plasma. Several studies have looked into the influence of blood cells on hemostasis and thrombin generation. Most research has been done on the influence of platelets on thrombin generation. Less is known about other cell types. During this presentation, I will shortly discuss about the effects of platelets, erythrocytes, white blood cells, and cancer cells on thrombin generation. And I will also show some results we have with a newly developed whole blood thrombin generation assay. So first, let's start with the platelets. So during hemostasis, coagulation from circulation bind to the platelet membrane receptors and to PS exposed on activated cells. This localizes the coagulation factors on the coagulant surface and enhances the enzymatic reactions. Additionally, thrombin that is formed during thrombin generation can also activate platelets by binding to the PAR1 and the PAR4 receptors. In the figure shown in the slide, not all platelet receptors and not all interactions are shown. So this is not a complete overview of all reactions on the platelet. It's just to illustrate some of the reactions. So the binding of coagulation factors to receptors on the platelet membrane influences thrombin generation. Blocking of membrane receptors, such as alpha-2b beta-3 with abciximab, or inhibiting PS exposure with annexin 5, decreases the peak and the endogenous thrombin potential in platelet-rich plasma. Deficiencies in memory receptors, such as a deficiency in GP1B alpha, as is seen in the Bernard-Soulier syndrome, or deficiency in alpha-2b beta-3, as is seen in Glanzmann's thrombasthenia, also leads to reduced thrombin generation in platelet-rich plasma. Apart from receptors and PS exposure, platelets also have granules. And there are two types of storage granules, the alpha granules and the dense granules. The content of these granules is released upon activation of platelets. The dense granules contain, among other, polyphosphates, which enhance the activation of factor 5 and factor 11 by thrombin. The alpha granules contain several procoagulant factors such as prothrombin, factor 5, fibrinogen, von Willebrand factor, and factor 13, but also anticoagulant factors such as TFPI and protein S, and also factors that have an effect on the APC resistance, like platelet factor 4 and also activated factor 5. The influence of the platelet granules on thrombin generation is apparent from the observed differences between thrombin generation in platelet poor plasma and platelet-rich plasma in patients with factor 5 deficiency. Some patients with factor 5 deficiency do not have life-threatening bleeding, and this is believed to be because in these patients, factor 5 from platelets is able to support thrombin generation in platelet-rich plasma, whereas if we look in platelet poor plasma, there is no thrombin generation. Additionally, in patients who have a defect in the alpha granule protein contact, a disease called Quebec platelet disorder, patients have a normal thrombin generation in PPP, but a reduced thrombin generation in PRP, and these patients have a bleeding tendency. Also, the platelet numbers and the volume of the platelets influence their effect on thrombin generation. We see that if you have an increased number of platelets, but also an increased volume, the lag time of thrombin generation is reduced and the peak is increased. So they're more procoagulant phenotype of platelets. Additionally, platelets can interact with von Willebrand factor. And there are two ways in which von Willebrand factor influences thrombin generation. Well, it is a carrier for factor 8, and hereby it brings factor 8 to the site of injury. Additionally, the interaction of von Willebrand factor with platelets induces platelet adhesion and aggregation. And the thrombin generation in the presence of platelets is also sensitive to the activation status of von Willebrand factor. And finally, platelets are able to release microparticles which are PS positive, and this enhances thrombin generation. Some research has been done that focuses on the influence of erythrocytes on thrombin generation. And increased hematocrit as well as abnormal erythrocytes have been associated with the occurrence of thrombosis. It's believed that the risk of thrombosis increases with an increase of hematocrit because there is a subset of erythrocytes which which are positive for PS, which express phosphatidylserine on their membrane. It's believed that around 0.5% of erythrocytes are PS positive. And this PS is able to support thrombin generation and hereby increase the peak of TG. It's also believed that it could be an anticoagulant effect because on the membrane of the erythrocytes, the activation of prothrombin goes through the formation of mesothrombin, which is able to support the activation of protein S, but doesn't cleave fibrinogen to fibrin. However, overall, we see that an increase in hematocrits mostly leads to an increased risk of thrombosis. Therefore, the effect of the PS exposure in supporting thrombin generation is probably more important than in supporting misothrombin formation. When we look at the red blood cell disease, sickle cell disease, we see that in the patients, there have up to 10-fold increase in their PS exposure on red blood cells. So they express more PS on their membranes, and this is only visible when we measure thrombin generation in whole blood, but not when it's measured in platelet pool plasma. So it is really the sickled erythrocytes that increase thrombin generation in these patients. Also, erythrocytes and platelets are able to interact, and they interact via receptors on their membranes. So the interaction of the ICAM4 on red blood cells and alpha-2b beta-3 on platelets leads to the activation of platelets. The interaction of the Fas receptor on erythrocyte and the Fas ligand on platelets leads to PS exposure on both platelets and erythrocytes. So these interactions between the platelets and erythrocytes both increase the ability to generate thrombin. The influence of white blood cells on thrombin generation is largely unexplored. However, some research has found that monocytes is able to synthesize and express tissue factor on their membrane and are able to release tissue factor positive microparticles in response to various cytokines. Neutrophil extracellular traps, NETs, are released from neutrophils after exposure to, for example, microorganisms, cytokines, or activated platelets. And several components of NETs have been shown to trigger coagulation, for example, by triggering autoactivation of prothrombin into thrombin. And activated leukocytes induce the activation and aggregation of platelets through granular release and interaction between the platelets and the leukocytes. So if we look into cancer cells, which are normally not present, of course, in healthy individuals, we see that patients with cancer have a 5-fold increase of developing venous thromboembolism, and this risk increases up to 28-fold for hematologic malignancies. The mechanism is not completely understood. However, several cancer cell lines have been shown to express tissue factor, and these cancer cells are also able to support thrombin generation in platelet pool plasma. Additionally, cancer cells produce tissue factor positive and PS positive microparticles, which enhance thrombin generation. And tumor-derived factors such as ADP, thrombin, and cytokines can directly activate platelets and hereby support thrombin generation. So now we've discussed the different types of cells and their influence on thrombin generation. So now we will continue the presentation and talk more about the different assays. So first, in the past there were 2 whole blood thrombin generation assays which have been developed that have been used in several research articles to look into the influence of cells on whole blood thrombin generation. In the first method, the whole blood coagulation assay is a subsampling method in which the formation of thrombin-antithrombin complexes is measured to determine thrombin generation. So at defined time points, a subsample is taken of the blood. The reaction is quenched with a cocktail of inhibitors, and then thrombin-antithrombin complexes are measured. The main disadvantage of this method is that it's very labor and time consuming. Furthermore, approximately 10 years ago, we also developed a CAT-based method for whole blood. And in this method, the porous matrix was used to create a thin layer of blood, and this resulted in the entrapment of the erythrocytes. However, the porous matrix also triggered contact activation and thereby influence also thrombin generation. And that's why we decided to develop a new whole blood thrombin generation assay, also based on the calibrated automated thrombogram method, but without the need of a porous matrix. So to prevent the erratic signal from erythrocyte sedimentation, which in the porous matrix assay method was prevented by forming a thin layer, we now continuously move the plate during measurement. And as comparable to the CAT assay in plasma, we have a calibrator well and a simultaneously measure a thrombin generation well to allow the calculation of the amount of thrombin that is generated during the TG assay. So what we do is we mix citrated whole blood with the substrate and a trigger mix containing tissue factor and calcium. And after vigorously mixing all these the whole blood with these reagents, we transfer the blood into the measurement plate. And then fluorescence is continuously measured comparable to the CAT assay in platelet pool plasma. And this assay was published last year in JTH. So our assay is sensitive to the count of platelets. That's, as you can see from the figure, if you increase the amount of platelets present in the assay, you see that the lag time is shortened and that a peak of thrombin generation and also the amount of thrombin generation is increased. And we also added synthetic phospholipids. This is seen in the figure on the right to the assay. And then we see that in the presence of synthetic phospholipids at the same concentration that is used in the plasma PVP CAT, there we cannot see any influence of platelets anymore. And it's even so that the reaction is even enhanced compared to reaction with a physiological level of platelets. And so this really highlights the importance of using the individuals platelets when we want to see what is the influence of platelets on thrombin generation. Because in the PPP assay, the phospholipids really trigger thrombin generation even more than the regular platelets. Additionally, to see the effect of platelets, we also used an activator of platelets, convolxin, and then we see that we have a faster onset of thrombin generation. And if we inhibit the platelets, we see a reduced thrombin generation. So the lag time is increased and the thrombin peak is lower. And it's especially when we used RioPro, an alpha-2 beta-3 agonist, this was most pronounced. So next, we also investigated the influence of erythrocytes in the presence of platelets on thrombin generation. And here we show that if we increase the amount of erythrocytes, that it also increases the peak of whole blood thrombin generation. And as you can see on the figure on the right, however, erythrocytes do not have any influence on the lag time, as can be seen on the figure on the left. So this is different from the platelets, which both influence the lag time and the peak of thrombin generation. So next we investigated the potency pathway in whole blood. So and therefore we added thrombomodulin to our reaction. And thrombomodulin is a cofactor in the activation of protein C by thrombin. And activated protein C is able to inhibit factor 5A and factor 8A and hereby reduces thrombin generation. So we performed the whole blood thrombin generation assay in the presence of thrombomodulin in a healthy population. And here we observed that the inhibition of thrombin generation by thrombomodulin was correlated with the erythrocyte count in whole blood. And when the red blood cell count increased, we saw less inhibition of thrombin generation by thrombomodulin. And these observations were done in healthy individuals, so with normal hematocrit. So probably, if you would measure in a disease group, which has also not only maybe different hematocrit, but also diseased erythrocytes, the effect on the APC pathway could even be increased. So we further investigated this observation using whole blood thrombin generation and PRP thrombin generation. So first, we determined that the erythrocytes had a similar inhibitory effect on activated protein C as it has on thrombomodulin. So the influence of the erythrocytes is not on the conversion of protein C to APC. And additionally, we saw that both for APC and for thrombomodulin, the inhibitory effect was 2 to 3-fold weaker in whole blood compared to platelet-rich plasma. And for these experiments, the PRP and the whole blood we used was from the same donor. So it's the differences are not related to any of the coagulation factors. So we showed that the inhibitory effect of erythrocytes is also dose dependent. As you can see here in these figures, if we increase the erythrocyte count, we see that there's less inhibition of the ETP and the peak by APC and by thrombomodulin in our whole blood thrombin generation assay. So we are still investigating the mechanism for this inhibitory effect of the red blood cells on the protein C pathway. And finally, I would like to show you some results we had in our patient groups with patients with multiple myeloma, which is a hematologic malignancy. And cell-mediated thrombosis is an important cause for morbidity and mortality in cancer patients. And cancer and also its treatment are all both well-recognized risk factors for development of thrombosis. And so the risk, the absolute risk depends on the tumor type, but also the stage of the cancer and treatment. But for the hematologic malignancies, the risk of developing cancer is 28-fold higher compared to people without cancer. So in multiple myeloma, due to the overgrowth of cancerous cells in the bone marrow, the level of normal blood cells decline. So patients have low levels of platelets, white blood cells, and red blood cells, and they also have an increased risk of bleeding, but also of thrombosis. So we performed a case control study in which we measured thrombin generation both in whole blood and in platelet pool plasma. So the two figures on the left are thrombin generation performed in whole blood, and the two figures on the right are thrombin generation performed in platelet pool plasma. And as you can see, in whole blood, we saw that the lag time of thrombin generation was increased in patients. So there was a delay in the start of thrombin generation. But we also saw that the ETP was increased in patients compared to controls. So the amount of thrombin that's formed was also increased. So whereas when we look in platelet pool plasma, we did not observe a difference between our control group and the patients. So to summarize, we saw in whole blood that there was a longer lag time and an increase in ETP. But when we looked in thrombin generation, there was no, in platelet pool plasma, there was no difference between patients and controls. So both the onset of thrombin generation is delayed, but the potential to generate thrombin was increased in whole blood of patients with multiple myeloma. So there are some limitations to using thrombin generations in the presence of blood cells. There's still the influence of flow and of the endothelium is not taken into account, and compared to thrombin generation in platelet pool plasma, it's more difficult to standardize. Measurements have to be done on the day of blood collection because it's cannot be stored. So these are limitations of blood cell thrombin generation assays. However, I hope that I've been able to show you that it is important to also, at least in some cases, to include blood cells in thrombin generation, because the blood cells and the interaction between these cells are important players in coagulation, and several disorders cannot be picked up in the absence of cells. So yeah, we think that whole blood thrombin generation is a new opportunity to study the involvement of blood cells in coagulation, and that there are several pathological conditions where the patients have both disturbed blood cell count, but also a higher risk of thrombosis. And we hope that whole blood thrombin generation may be the bridge to link both these disturbed blood cells and the risk of thrombosis. Thank you for your attention.